Welcome, everyone, to our webinar series. We have an industry partner update with Avidity Biosciences. I think the majority of you already know, but we're Defeat Duchenne Canada. We're the country's only national charity dedicated to ending Duchenne muscular dystrophy. We have a goal to provide leadership in research, advocacy, and support to ensure every Canadian affected by Duchenne can live a long, active life. We envision a future without Duchenne. And we have 4 strategic pillars that we focus on: research, advocacy, education and support, and community engagement. Today, as I mentioned, we're pleased to welcome you to this webinar with information from Avidity Biosciences. We have 3 people from Avidity who are going to be presenting. We have Erica Escartin, who is the Senior Director of Global Patient Advocacy. We have Lisa Ackerman, the Senior Vice President of Clinical Development, and Hussam Yunus, who is the Global Program Head. I'm going to now pass it over to, I believe, Erica, you will be presenting first.
Thanks so much, Judy, and I'm going to share my screen so that everybody can see this okay. And hopefully we can. Hold on, let me just put this in. Perfect. Thank you, Judy, for that kind introduction and welcome. And the Avidity team is very happy and pleased to be partnering with Defeat Duchenne Canada to walk through the initial data from the EXPLORE44 clinical trial, the evaluation of del-zota for the treatment of individuals with Duchenne muscular dystrophy, mutations amenable to exon 44 skipping. Avidity is a publicly traded company, and as part of being a publicly traded company, we always have to make sure that everybody is aware during the course of our presentation that we will be making forward-looking statements. These statements are on our website, and you will see the full paragraph of all of them in detail. But essentially, a forward-looking statement is a term defined under applicable law and allows us to say things based on what we know and our current beliefs, even though they're not guaranteed and can change due to factors within and beyond our control. In this presentation today, we will include forward-looking statements, including but not limited to the characterization of del-zota, the EXPLORE44 trial, design and goals, and our expectations for regulatory interactions related to the EXPLORE44 trial. I won't go into detail into the other bullets, but if you are interested in reading about them more, you can go to our website as well. So at Avidity, our mission and our vision since the company was founded today and even through the future is to profoundly improve people's lives by revolutionary the delivery of RNA therapeutics. We are a biotech company based in San Diego, and we are truly grateful and honored to serve the Duchenne community and consider, patients truly at the heart of everything that we do in our daily activities at Avidity. We are obviously very grateful and very excited to be here today with you, and also for the partnership with the Defeat Duchenne Canada group, and also ensuring that we're partnering and providing all the essential information throughout our program development. We are here today because of you and together with you. And so with that, I'm going to pass it over to Lisa, who's going to walk us through the initial data of del-zota. So Lisa, over to you.
Great. Thank you, Erica. So for those of you who are familiar with our DMD program, you might have heard us call our drug AOC 1044, but we've actually been given a generic name by the World Health Organization, and that name is delpacibart zotadirsen. And the delpacibart refers to the antibody part of the drug. And if you see that figure on the left hand of the slide, that's a figure of the drug, and the antibody is depicted in the purple and bluish colors. And the zotadirsen is the word for the PMO or the oligo that is attached to the antibody. And the oligo is shown in those little green squiggly lines. Because it's such a mouthful, we have shortened the name to del-zota, and that's the way we'll be referring to it throughout the presentation. Okay, if you can go to the next slide. So how does del-zota work? As I said, del-zota is a drug that includes a monoclonal antibody and PMOs that are attached to it. The role of the antibody, it just has one thing that it has to do, and that is to bring the PMO into the muscle cell. And it does this by binding to a receptor that sits on the outside of muscle cells called the transferrin receptor. When the antibody binds to the transferrin receptor, it is transported inside the cell. Once it gets inside the cell, the PMO or oligo pieces come off, and they make their way to the nucleus to do their job. And their job is to bind to the dystrophin RNA and cause the cell machinery to skip over exon 44. This allows the cell to produce dystrophin protein. So if you go to the next slide. Okay, so today I'm going to show data from our preliminary assessment from our trial called EXPLORE44. And the next slide shows the design of this trial that we're talking about today. So there were two parts. Part A was in healthy volunteers, and Part B was in DMD participants who have mutations that were amenable to skipping exon 44. So in Part D and Part B, we are evaluating two doses, 5 mg per kg and 10 mg per kg. Now, this is a double-blind, placebo-controlled trial, which means that some of the participants receive placebo and some of them receive the active drug, or del-zota. In the 5 mg per kg cohort, patients receive either placebo or del-zota by IV infusion 3 times every 6 weeks. In the 10 mg per kg cohort, patients receive either placebo or del-zota 3 times by IV infusion, but every 8 weeks. After completing the Part B of the study, the participants were eligible to go into our extension study, which we call EXPLORE44 OLE. And in this extension study, everybody receives active study drug. Now, this study is ongoing. We haven't finished it. So today we're just going to be talking about data from our lowest cohort, which is the 5 mg per kg cohort. So if you go to the next slide, this slide just gives a little bit more information about some of the key design for this trial. Now, Part B was designed to enroll 24 DMD participants with ages ranging from 7 to 27. It enrolled both ambulatory and non-ambulatory participants, and biopsies were collected both at baseline and after those 3 doses. Now, the biopsies were really important because it allowed us to see how much of the study drug got into the muscle tissue, and also to see how much exon skipping and dystrophin there was. But the primary objective of the study was safety and tolerability, and that's always the primary objective of these phase 1 studies. The secondary objectives included pharmacokinetics, and that just means how the drug is distributed in the body and how it is broken down. And like I said, other important secondary objectives were exon skipping and dystrophin levels. Now, in this study, we also measured some measures of muscle function, and we also had a number of patient questionnaires and quality of life measures. But we're not going to talk about those exploratory objectives today. We're just going to focus on our primary and our secondary objectives. So if you go to the next slide. So this is a slide that shows the demographics and baseline characteristics on the study. Remember, this was placebo-controlled. So we had 3 participants who received placebo, and we had 9 participants who received del-zota in the 5 mg per kg cohort. The ages, the mean or average ages were about 14 to 16 years of age. There was a good balance in the age of onset between the 2 groups, which was just less than 4 years of age. And we're showing here the baseline creatine kinase levels, which, as you probably know, in Duchenne's muscular dystrophy is quite high. In our placebo group, it was about 7,000, and in our treated group, it was about 5,000. And then everybody on the trial were on stable doses of corticosteroids. So if you go to the next slide. So as I said, the primary objective of the study was safety and tolerability. So this is a summary of our safety. There were no related AEs of the same type that occurred in greater than 1 participant. There was one participant who discontinued due to a serious AE of anaphylaxis, which fully resolved. And there was also one participant who discontinued due to a moderate infusion-related reaction. There were no symptomatic changes in hemoglobin. There were no cases of low magnesium, and there were no renal events on the study. So if you go to the next slide, please. All right, so that was the sort of like the background. So now I'm gonna show you data that were for our secondary objectives. So you can go to the next slide. Thanks, Erica. So the first step was to ask the question, was del-zota getting into muscle tissue as we had intended? And this is a slide that shows how much of that PMO did get into muscle tissue. And we're showing that in amounts of nanomolar, but what really matters, the main message is that there were significant levels of PMO detected in the muscle, and these levels were high enough to get exon skipping. If you look at that bar on the left hand of the graph, that is how much PMO we saw in the healthy volunteers, and that was only after a single dose. On the right hand is how much of the study drug we measured in the muscle tissue for participants who received del-zota, the DMD participants, and that was after 3 doses of study drug. So we clearly got the PMO into the muscle tissue. So the next question was there any exon skipping? So if you go to the next slide, this shows the results for the exon skipping. Now remember, we have 2 groups. We have placebo and we have our treated group. Now our placebo is shown on the left side of the graph in those gray bars, and the treated group with del-zota is shown on the teal bars on the right. So if you look at the placebo, we see some amount of exon skipping at baseline, but there was really no difference after receiving 3 doses of placebo as would be expected. On the right-hand side, you can see that the participants in the del-zota group had some baseline exon skipping, but after they received 3 doses of del-zota, that exon skipping increased on average by 37%. So there was a significant amount of 37% more of exon skipping on average that was seen in the del-zota treated group. All right, so we get in the muscle cell, we have exon skipping. So the next question we wanted to ask is, what did the dystrophin levels look like? And that's in the next slide. So again, the 2 groups are shown here, placebo on the left in gray, and the del-zota group in teal on the right. So the placebo group had some baseline dystrophin that was detected in the biopsies, but as expected, there really was no difference after the 3 doses of placebo. And on the right-hand side, again, you see some baseline dystrophin, but in the del-zota treated group, that increased on average 25% of normal. So that's what the whole group did on average. If you look at an individual patient, the increase in dystrophin was up to 54% of normal. So this was really nice to see that we saw very good increases of dystrophin after 3 doses of del-zota. So if you go to the final slide that I have here is on the creatine kinase. And so creatine kinase is can be a measure of muscle health. So it is released into the blood when there is breakage or damage of the muscle. And then you can measure it very easily because it's in the blood. And that's in Duchenne muscular dystrophy, there are very high levels of creatine kinase due to the ongoing muscle damage. As I said earlier, our placebo group had about 7,000 at baseline and our treated about 5,000. So you can see in gray is our placebo group over time. Their creatine kinase didn't really significantly change during the course of the study. But the del-zota treated group, you can see that it goes down over time during the study. And by the end of the study, there was about an 80% decrease in the amount of creatine kinase that was secreted into the blood in the patients who received del-zota. And these levels were actually near normal levels. So it was really great to see that the creatine kinase also decreased along with increases of dystrophin. So that's the end of all the data, and I'm going to turn it over to Hussam.
All right, thank you, Lisa. As Lisa nicely reviewed the data, I'm just going to summarize all of that here. So the AOC platform that we're working on here at Avidity really specifically delivers oligonucleotides to muscle, skeletal muscle tissue, as well as heart tissue. And as Lisa nicely showed, we got substantial amounts of the oligonucleotide for exon skipping in muscle tissue. That led to a substantial, statistically significant increase in exon skipping, exon 44 skipping, as well as dystrophin protein that's made from the newly formed mRNA transcript, a mean increase of 25% and up to 54% in the highest responding participant. And importantly, demonstrating a reduction in creatine kinase, which is a marker of muscle damage. So we're really excited about this initial data here at the 5 mg per kg cohort. Avidity is committed to moving this promising treatment forward. So we really appreciate and understand the responsibility to move this very quickly. In that respect, the FDA has granted del-zota orphan drug and fast track designation. And in Europe, we've also received orphan designation. Our focus at this point is to complete the EXPLORE44 trial, as well as the open label extension, which we have decided to increase the enrollment. And so we're adding additional participants in the open label extension portion of the trial. It's going to be important for us to really collect that data package to move this forward. And we are looking forward to interacting with regulators here in the US, specifically the FDA, to identify the most expeditious path for accelerated approval. So on behalf of the entire Avidity team here in San Diego and across the US, I really want to thank the DMD community. We couldn't have done this without their partnership, as well as the patients that have participated in the trials. Feel free to connect with us. The email is below. And Erica, I'll pass it off to you.
Sorry, I was on mute, and I was talking to myself. Thank you, Hosam. Got myself off of the screen share, and Judy, we can open it now to Q&A.
Absolutely. Oh, sorry, I'm having a technical difficulty. Just one moment. For some reason, there we go. So we do have some questions already. Here are the first couple, and I know that we also have some coming in as I speak. So with the results from exon 44 skipping, what is the plan for exon 51 skipping, and when could we see development of clinical trial on that aspect? Related, if you would be using the same methodology, would you expect to have a similar level of dystrophin expression for skipping 51 or 53? I'm not sure who would like to answer those questions.
Yeah, I can answer both of these questions.
Great.
So at this point, we're really focused on moving forward and collecting all of the data that we need for the exon 44 program, del-zota program. At the same time, we are working in the lab on the other exon programs. And at this point, we can't say too much about when we would anticipate those moving forward. But we are working on these in the lab at this point. In terms of being able to predict the potential activity relative to what we've seen already with del-zota. We know that the technology is really designed to deliver a lot more oligonucleotide to muscle tissue, and we've now demonstrated that. So compared to the naked PMOs, without that specific delivery to muscle, we do anticipate to see greater activity than those molecules. But to say exactly how much, it's difficult at this time.
Great. And then another question that we have was, when will del-zota be given to market in Canada? Don't know if you have any insights into that.
Thanks, Judy. I can answer that question. So as Hussam mentioned, our goal is to move as fast and expeditiously as we can in the US to get approval with the FDA on our program. And after that, we will assess our global regulatory strategy. But our goal is to get it approved as fast as we can with the FDA.
Great. And we have a couple of people asking about if you're opening clinical trial sites in Canada.
Hussam or Lisa, you want to?
Yeah, not at this point. So as Erica said, our goal right now is to finish the EXPLORE44 study and to engage with the FDA on our fastest path to accelerated approval. Once we have that understanding, we will be engaging regulatory authorities in other regions to understand what the clinical trial pathway would be for additional trials outside the US. And so more to come on that, but we don't have anything right now to share.
Sounds good. We have another question. Will trials include older people with exon 44 deletion? One of the people participating mentions their partner is 34 and typically excluded from clinical trial due to age.
Yeah, I know. So it's a very good question. And, our goal is to get del-zota approved for all ages of Duchenne, no matter what the limitation is for ambulatory and non-ambulatory participants. So right now in the clinical trial that we're running, that's ongoing, we did have an upper age limit of 27. So again, our focus is to finish that, and we'll see with other trials to come as we engage with the regulatories, what the exact age limitations will be for that. But our goal is to have it approved for all ages.
Great. Also being asked, if someone in Canada wants to participate at a US trial site, is that possible? And I'll add, if it is possible, are there certain sites that are more open to that?
Yeah, so that's a great question. And right now, as Hussam said, we are enrolling a small number of additional participants in our open label extension study. And the best way is just to contact the closest site near you and have a conversation with that site.
Great. Just checking to see if we have any other questions. I think that's all we have now. Oh, another one coming in, sorry. Another question is, realistically, what should be the expectation in terms of dystrophin expression?
Do you want me to pull up the slide again, Lisa, on the dystrophin expression?
Sure.
So we can show it again. Let me just... And I apologize for that black bar. I try. Let's see. Do you want to?
So yeah, so I think what we can say is the data that we have right now, which is in our 5 mg per kg cohort, and this is the teal bars on the right are the dystrophin levels that we measured after receiving del-zota for 3 times every 6 weeks. And we saw an increase of 25% on average. So that means that we took all those patients that we measured and we did a mean of them. Some were a little bit less and some were a little bit high. So I think that would be our expectation, the realistic expectation, that it would be somewhere in the range of a 25% increase from baseline at the 5 mg per kg dose level.
Great, thank you. We have another question. In the presentation, we talked about heart and skeletal muscles. Do we see improvements in the lung muscles as well?
So I don't know if Shusam, do you want to take that?
Yeah. So the data that we've shown today is skeletal muscle. We do have preclinical data that also demonstrates activity in heart tissue as well as diaphragm. So we believe that this should translate to those tissues as well. Obviously, that's very difficult to get at this point in the clinical trial. Anything else you want to add to that, Lisa?
No, that was perfect. Thanks.
Thank you. I have a follow-up question to the question about what should be the expectation in terms of dystrophin expression, which you had addressed. The follow-up question is the 25% sufficient to stabilize the condition?
So that's a really good question. And, we will be doing additional trials and following the patients who've been treated long term, so we can start looking at effects on some of the functional outcomes like muscle strength and ability to walk and those types of outcomes. And we don't have that data right now. But we can say is the hypothesis when we started this program was that we wanted to see an increase of dystrophin above 10%, because that is, you start getting in the level of a Becker dystrophin, in which it is can be a milder form of the disease. So with a 25% increase, we do think that we're in the range of the Beckers, and we are hopeful that that translates into slowing of disease progression or in benefit, but we still need to continue to follow the data. Hussam, did you want to add anything to that?
Yeah, no, that's exactly right, Lisa. I think it's important that we have a change of 25% absolute, but the average that these patients had at the 5 mg is around 32%. So that's kind of what Lisa's referring to in terms of above 10%. It's relative to that 32%. So we're substantially above that at this point.
Great. Another follow-up would be, are you anticipating any breakthroughs in the next 2 to 3 years?
Breakthroughs in terms of substantially moving quickly to get this drug approved as fast as we can. I think that's really our focus at this point.
And Judy, I know there was a question about how to find the sites in the US that would accept Canadian set clients or individuals. I added in the chat, in the Q&A, our EXPLORE44 study website, but I can also add it in the chat in the Zoom in case
That'd
people
be great.
do
Thank you.
that.
I appreciate it, Erica. We have a question as well. If people have been in other trials, are they still eligible for this trial?
So it really depends. I think the main thing that I would say is that we are not at this time accepting participants who have been on gene therapy trials. But other than that, it really is, we have exclusion criterion about the time from the last time they were in the trial before they can be eligible for our trial. So it's that besides gene therapy, it's just a matter of the time between the two.
Thank you. Does anybody else have any questions? I think we've completed what have come up during that time. I don't see any additional ones being asked. I'll give it maybe just a couple of seconds. I've really appreciated you all taking the time to go through this. It's been very informative and clearly, oh, we have one more. Let's see it's a thank you for everyone. So thank you very much, everyone, for going through it. Thank you to our participants. And I think we seem to have wrapped it up. Oh, one more question. My question about regulatory approval length of time wasn't answered. Do you have anything you'd like to address for that? Even maybe timelines for submissions to different regulatory bodies. I suspect it's early for that, maybe.
Yeah, I think right now we need to, and we're working on engaging with the regulatory authorities. And until we've had those conversations and we understand our path forward, it's too early for us to speculate on timelines. But we will, as soon as we understand what they are and we do know our path forward, we will definitely be communicating that to the community.
Wonderful. Well, thank you, everyone. Thank you to our participants for all your excellent questions. I've just put up links to where you can find us as well. Thank you to our panelists for coming on and taking the time to go through your exciting trial results. We look forward to your next update.
Thanks so much, Judy, and thank you for everybody that joined today. Have a nice afternoon, everybody.
Thank you, you too.